Hello, our group presentation is on Retroviridae. I'm Mixley.
I'm Diana.
I'm Vanessa.
And I'm Brian.
Retroviridae. There are two types of retroviruses, delta retroviruses, HTLV1 and HTLV2, and lentiviruses, HIV1 and HIV2. The retroviral capsids are closed protein shells that adopt heterogeneous morphologies such as tubular, conical, nearly spherical, or polyhedral, and their extended host range includes rodents, cats, dogs, monkeys, and human cells. It contains a ssRNA plus genome and can cause tumor growth and certain cancers in animals. In humans, on the other hand, a retrovirus known as HTLV1 causes a form of cancer called adult T-cell leukemia. The retrovirus known as HIV causes acquired immunodeficiency syndrome known as AIDS. The HTLVs are endemic in Central Africa, the Caribbean islands, and in Southwest Japan, but now tend to spread through the IV drug user population in the US and in some countries in Western Europe. The genome. The genome consists of the reverse transcription of the viral RNA genome into DNA, degradation of the RNA, and integration of the double-stranded HIV DNA into the human genome. The 5' LTR region codes for the promoter in the order to begin transcription of the viral genes. The reading frame of the GAG gene goes from the 5' to 3' end and encodes the proteins of the outer core membrane, the nucleocapsid, the capsid protein, and the small nucleic acid stabilizing protein. It also includes the pol reading frame coding for the enzymes protease, reverse transcriptase, and RNase H, RT plus RNase H, and integrase. Besides its structural proteins, the HIV genome codes for several regulatory proteins such as transactivated protein and RNA splicing regulator, which are crucial for the initiation of HIV replication. This can be shown in the picture. The other regulatory proteins, such as negative regulating factor, viral infectivity factor, virus protein R, and virus protein unique, affect viral replication. Virus budding and pathogenesis, HIV2 codes for virus protein X instead of virus protein unique, which is instrumental for the reduced pathogenicity of HIV2.
There are 7 stages of HIV cycle. The first stage is binding. During this process, it attaches itself to receptors located at the top of the CD4 cell. The second stage is fusion, which is when the HIV envelope and CD4 membrane fuse in order to enter the cell. The third stage is reverse transcriptions. And during this process, HIV uses reverse transcriptase to convert its genetic material, HIV RNA, into HIV DNA. The fourth stage is integration, which is when HIV releases integrase. It is then used to insert its DNA into DNA of the CD4 cell. The fifth stage is replication, where HIV then starts making long chains of HIV proteins using machinery of the CD4 cell. The sixth stage is assembly. It is when HIV proteins and HIV RNA go to the surface of the cell and form a new immature HIV. After that, it's the seventh stage, budding, and it is when a new immature HIV exits the host cell, then releases the protease. The protease then breaks up the long protein chain, which creates the mature infectious virus. Pathogenesis. HIV pathogenesis involves a chronic infection which involves a slow degradation of immunity that will eventually lead to AIDS. HIV is a virus that causes AIDS by having contact with a big amount of different types of cells in the body. It is transmitted mainly through blood and genital fluid from another person that has HIV, or it can be passed from a pregnant mother with HIV to its newborn baby. The steps to infection of HIV don't only consist of it interacting with CD4 cells, but also coming in contact with many other cellular receptors. There is a variety of intracellular mechanisms that determine if it is leading to a productive or latent infection.
Now that we have gone over the life cycle and pathogenesis, I will be talking about how HIV is transmitted and its risk factors. HIV is transmitted through sexual contact, receiving blood transfusions that have been contaminated with HIV. It can also be passed down from a mother to a baby during pregnancy and breastfeeding. Sharing needles and injection equipment can also lead to HIV transmission. People are more vulnerable to get HIV by having unprotected sex, which is one of the most common risk factors. Another risk factor are unsanitary needles, which increases the chance of transmission. Next, I will be discussing the symptoms of HIV. People do not typically show symptoms right away because it takes about 2 through 4 weeks to appear. There are 3 stages of HIV infection. The first one is acute HIV infection, and symptoms are flu-like. For example, people may have chills, fatigue, rashes, sore throat, and so on. These symptoms usually last for days or even weeks. The second stage is chronic HIV infection, also known as clinical latency. During this stage, there may be no symptoms or infection. The third stage is acquired immunodeficiency syndrome, AIDS. Some of the symptoms are depression, rapid weight loss, chronic diarrhea, recurring fever, memory loss, and so on. Next, I will be talking about HIV and how it could be detected and the treatments for it. There are 3 types of tests one can take in order to see whether they have HIV or not. The tests are nucleic acid test, antigen antibody test, and antibody test. These tests are typically performed through oral fluid, blood, and urine. As of now, there's no cure for HIV. However, with medication, it can be controlled and further complications can be prevented. A treatment for HIV is called antiretroviral therapy. The most effective kind is one that uses a combination of different HIV medicines. There are 7 drugs of HIV medicines, which are NNRTIs, NRTIs, PIs, fusion inhibitors, CCR5 antagonists, INSTIs, post-attachment inhibitors. Antiretroviral medicines have to be taken daily in order to help reduce the growth of the virus and transmission.
To understand the evolution of HIV, it is important to look into the history behind it. That being said, while researchers were investigating what made AIDS infectious, other scientists realized that monkeys suffered from a similar disease. Therefore, they decided to take a blood sample from these monkeys, and in 1985, they discovered that it was simian immunodeficiency virus, or SIV for short. Because this virus was similar to HIV, researchers started to hypothesize that HIV originated from our primate relatives, crossing the species barrier. With further investigation, researchers found that HIV was even more closely related to the chimpanzee's version of SIV, thus were the ones who passed it to humans. Where it originated has been controversial, but most scientists agree that it came from West Central Africa in the early 20th century. So you may be wondering how this spillover came to be. Well, the cut hunter hypothesis is a potential answer. In the early 20th century, hunters in West Central Africa had a taste for bushmeat, specifically monkeys and chimps. While butchering these infected SIV animals, blood would sometimes enter either through the mouth or exposed wounds. Again, this is just a hypothesis, but it is one of the most respected ones. The virus then spread from the African continent around the 1960s through a single carrier, eventually making its way around the world. HIV became an epidemic because of how quickly it can mutate. This virus was able to evolve and alter its genetic sequence as a result of replication and recombination. As mentioned earlier, HIV stores its genetic information in the form of RNA, and because of this, it has to be converted into DNA in order to incorporate its genetic information in the host cell. However, each time it does this, the produced sequence may be slightly different than the one prior, and over time, mutations can accumulate, giving the HIV virus new characteristics. As for recombination, reverse transcriptase can cause a process called template switching. So if a cell is infected by 2 strains of HIV, reverse transcriptase can combine the 2, ultimately accelerating the production of new variants, which in turn causes drastic changes. This image on the right shows a phylogenetic tree of HIV, specifically of HIV-1 recombinants and the distinct subtypes. As mentioned earlier, this virus mutates quickly. Therefore, scientists to this day struggle to understand every component of HIV, which is why we see many lineages lacking information. Despite this, we can see the spillovers through the branching of this tree. Additionally, we see complex recombinant patterns and the observable diversity of subtypes as indicated by the worldwide map. With that being said, this concludes our presentation.